Okay, so first I would like to thank the organizers for giving me the opportunity to share with you some of our recent data. In the picture, you can see the two people who actually did the work. One is, the first is Ronen Blecher. Both of them, as you can see, the pictures are orthopedic surgeons. Ronen is being now in Assuta Ashdod, working as a physician, and Ran Asraf, which recently finished his PhD in my lab, is now in Asaf HaRofeh. Okay, so so for many years, my lab was interested in trying to understand developmental biology or musculoskeletal developmental biology. And mostly we were interested in understanding the role of muscle or muscle contraction, or the force that muscle contractions are basically extracted on the skeleton. And in a recent review, in a review that we wrote a few years ago, we summarized a lot of the details from our lab and others. And we found that muscle force really regulate many aspects, including chondrocyte proliferation in the growth plate, column formation, and chondrocyte intercalation. When we talk about bone morphology, muscle force regulating the morphology of the bone, and also if we talk about bone eminence, these protrusions that coming out of the skeleton and where tendon and muscle bind to the bone, we found that if you have no muscle force, that basically these eminence are not forming. And finally, we and others show that you need muscle and embryonic movement in order to have joint formation. And this is not obviously true for mice. We all know that embryos move quite a lot, as you can see in this movie. And there is a syndrome called fetal akinesia deformation sequence, which gather many syndromes that ending up in restriction of embryonic movement, causing to a variety of problems, some of them related to abnormal skeleton development. So what is important to say about that is that in all this work that we did at that point, we used either mutants that failed to have muscle formation or paralyzed ones. So namely no movement whatsoever. So now I want to go to the next slide, but it doesn't work. So 1 second. Oh, yeah. Okay. So what I want to talk now is about the importance of muscle regulation in two different aspects. One is spine, and the other one is joints. In the spine, I don't know if any of you ever thought about what actually keeps the spine straight. As you can see, it's a structure that built from repetitive vertebra separated by joints. And it's really interesting to understand what keeps it straight. Obviously, this is not just the philosophical questions. It's actually manifested in a devastating syndrome called adolescent idiopathic scoliosis. As you can see here in the X-ray, where there is a severe loss of the normal structure. Another interesting question that one can ask is the proportion or growth of proportion. And that is, if you have a joint forming in the embryo, obviously as the organism is growing, the morphology of the joints and the proportion of the joints needs to maintain. And what is the mechanism that regulate that is still an open question. However, again, this is not just a philosophical question. It is manifested in another syndrome called DDH, or dysplasia of the hip. And what is important to say is that these two phenotypes, both these two syndromes, scoliosis and DDH, are by large the two most common syndromes that known for children. And so going back to the muscle, thinking about these two problems, we start thinking that maybe muscle or muscle force regulating these two, involved in regulating these two aspects. So for instance, what if the muscle that are stretched along the spine basically regulate its right alignment? And what if the movement or certain movement that the organism is acting or forming is important to regulate the morphology of the joint? Now, obviously, you cannot come up with the strategy of losing or inhibiting muscle activity to ask these questions, but actually you need a more another more sensitive or delicate system. And for that, we thought that maybe the proprioceptive system might be our system. And I'm not sure that many of you actually know what it is. I wasn't aware much to that system before. So just a crash course on proprioceptive system. So the proprioceptive system is actually a system that allows us to understand where our extremities are located in space without seeing them. So if you close your eyes and you move your hands, you know exactly where they are. And we capable of doing so simply because we have inside our muscle two kinds of receptors, one in our belly. When I say receptors, I'm not talking about proteins now, I'm talking about organs, small organs. So in our muscle body, we have one organ that's called muscle spindle. And in just between muscle and tendon, we have another organ that's called Golgi tendon. One feel muscle stretch and the other one feel muscle tension. And once there is a change in the length or tension of this muscle, they send, there is a signal that runs through sensory neurons into the spine, where in one case it forms a synapse with a motor neuron that goes back into that muscle and basically control its length or movement. And that's known as the monosynaptic stretch reflex circuit. And concomitantly with that, the information is running to our brain, where the proprioceptive picture is forming. And proprioception is very important for regulating our movement. And if you don't have proprioception, your movement, you're becoming ataxic. So what we're looking is and interesting to say that there is not a lot of molecular information about this system. So we start looking for some genes that might be regulating them. And what we found is that a gene called RUNX3 is very important for the surviving of the TREC-C proprioceptor neurons that going from the Golgi or from the spindle to the spine. And if you now look on RUNX3 deficient mice, using CT, what we found was the fact that they developing severe scoliosis, curvature of the spine. As you can see over here in this graph, this is a control. It's close to 20 animals. All of them, the spine is aligned, as you can see on the 0 axis. And then in the RUNX3, you can see that most of the animals curve, and the curve is to the right. And this is an important point. Now, one thing, why it is important? It is important because in humans who have scoliosis, they turn to the right as well. Now, one thing that I didn't mention was the fact that adolescent idiopathic scoliosis, the kid's bone normally, it's not a congenital disease, and just around their puberty, they developing these, they start to lose their spine alignment. So we were interested to know whether in RUNX3 mice, the scoliosis is acquired or congenital. And for our surprise, we saw that if you look at these, the spine of these animals around day 40 postnatal, the spine is completely straight, as you can see over here. And as they getting closer into puberty, they start curve to the right, similar to the humans. So one thing which is important to remember or important to say right now is that the fact that until we identify this phenotype, scoliosis was supposed or was believed to be phenotype of only bipedal, only animals to walk on two, and not especially not acquired one, and not not animals that walk on four like mice. Okay, so one of the things that I'm sure that run through the brain of all of you is that, okay, but RUNX3 is a major transcription factor. He has a brother called RUNX2, very important for skeleton formation. And also there were some publication, RUNX3 was important in bones as well. So we decided to take a conditional knockout approach, and we knocked out RUNX3 in osteoblast using the COL1, and we didn't see any phenotype. We knocked it out in cartilage using COL2 CRE, no phenotype. And then we said maybe it's important in the tendon, so we knocked it out using sclerosis CRE, which deleting genes in the tendon, and again, no phenotype. So that basically led us to think that indeed what we're seeing here is a specific role of RUNX3 in proprioceptive neurons. And to take it further and also to trying to bring it into more situation where it will fit into a human situation, because it's hard to believe that a human that will lose RUNX3 gene, his problem will be that he develops scoliosis. Obviously, RUNX3 is involved in many other very important aspects of development, and that won't be the problem. Luckily for us, we took advantage of the fact that a lab in the Weizmann, the Yoram Gruner's lab, were studying regulation of RUNX3, and they identify 3 specific enhancers that regulate the expression of RUNX3 in TRAC-C proprioceptive neurons. And since RUNX3 is important for their survival, they basically using CRISPR-Cas knocked out different combination of these enhancers and check for the proprioceptive survival. And you can see here in the different combination, you lose almost all the TRAC-C neurons if you eliminate these 3 enhancers. So it's important to remember these mice have RUNX3 intact, they're just missing the enhancers. So we thought, okay, let's look on what's going on with these mice with this scoliosis, and you can see that different combination leads to different develop of different angle of scoliosis. So that was, again, it's important to say, with these mice, you lose just the enhancers that push the expression of RUNX3 in proprioceptive neuron. The rest of the expression in other organs is normal. And finally, just to get out of the RUNX3 and to ask, because I told you that RUNX3 important for the TRAC-C neurons that coming both from the Golgi and from the spindle, we wanted to ask whether losing even one proprioceptor will lead to scoliosis. We found paper describing another gene called EGR3, which is important for the genesis of muscle spindles. So in this case, in this mutant, you just lack the spindle, you still have the Golgi. And again, we got scoliosis again acquired. However, I have to say that the amplitude wasn't as high as the RUNX, suggesting that you need the activity of both proprioceptors, the Golgi and the spindle. Right. So we summarize all that in a paper that basically claimed that RUNX3 and EGR3 mutant mice are a new model for scoliosis with the closest resemble to the human condition. We suggested that adolescent idiopathic scoliosis have a neurological side or may be thought as a neurological syndrome. Whether you're interested in the human condition or not, I think that it's really a very exciting example showing that the proprioceptive system play a key role in spine alignment. And finally, I think it's an interesting direction to think about muscle as acting as a sensory tissue that play non-autonomous role in skeletal integrity. So when we finished that, another thing that came into our mind was the fact that basically, unlike maybe what most people would think, if you think about the etiology of human syndromes, namely, if you have a problem, if you have a skeletal syndrome, you look for problems in the skeleton. What we identify is that regulation of muscle behavior by the proprioception regulate or important for a key aspect in the skeleton or skeleton integrity. And that brings up the possibility that maybe there are many others musculoskeleton syndromes that their etiology lay in misfunction of the proprioception. And for that, obviously, one would like to know more about the molecular mechanisms or the genetics of these systems. Interestingly, for some reason, although there was, when molecular biology and genetics, molecular genetics revolution happened, and we know that many organs, their molecular components and genetics were identified, and obviously in neurology, many of the key molecules have been identified. For some reason, the proprioceptive system left behind. Although now, just recently, in the last year, there is an attempt for few groups to fill this gap. Still, the key player are left behind. And we decided to take on ourselves this challenge, and we isolated muscle spindle from the mastication muscle in mice. And as a control, we took the muscle where they're embedded and we ran their transcriptome, and as you can see from this heat map, we got a nice clusters showing that we got some specific genes, and actually we got like 3,000, more than 3,000 specific genes that are differentially expressed between muscle and muscle spindle, and this is a Volcom plot showing you the different genes, and we start looking for these genes, and we concentrate on a specific family that called mechanosensing ion channels, simply because we were looking for molecules that can translate mechanical stimulation into molecular signals. And we got some, and this is just a partial list of it, and one of them was piezo2. And the reason why we were so excited was simply because there was a paper that published a few years before showing that piezo2 is a principal mechanotransduction in the proprioceptors, and this is just showing you it is expressed both in Golgi and in muscle spindle. And that basically provide us with an opportunity to check our main hypothesis that by identifying genes that are expressed in the spindles, we can go and check what's going on with the musculoskeleton, and whether these genes might regulate different aspects in the musculoskeleton. Not only that, there was another reason for our excitement, because if you
you have 1 minute.
What I have?
1 minute, please.
Yeah, it's okay, no problem. Because we were able to, there was a publication 2 or 3 years ago showing that mutations in human, in piezo, ended up in some skeletal phenotypes like scoliosis and hip dysplasia. Obviously, in human, you don't know in which tissue you need the piezo function. So we decided to go ahead and knock out piezo just in the proprioceptive system. And when we knocked it out just in the neurons of the proprioceptors, we got scoliosis of the spine and kyphosis. And again, just because piezo, I don't know if you follow the literature of this gene, but it is expressed in many others, including in the skeleton. So we knocked it out, both in using COL1 again for osteoblast or COL2 for chondrocyte, and we got no phenotype whatsoever. Now, you remember that these patients also had some problem with their joints. Now, I'm not going to go through all the details because it's a bit complicated. Just trust me on the fact that there is ways to measure different ratio that tells you how well the femoral head sits inside the acetabellum in the hip, and by measuring these numbers, you can basically conclude how well is the fitting between the two. And we were able to see that there is a problem in the hips of these animals, namely there is no well adjustment. And again, if you do that, if you lose piezo now in mesenchymal cells, which will give rise to either cartilage or bone, you have you cannot recapitulate this phenotype, suggesting that, again, the effect of piezo in the proprioception leads to two major phenotypes in the skeleton, scoliosis and abnormal hip adjustment. And just to make the point, just to finish the point, if indeed the proprioception is important, then obviously RUNX or EGR should show or should have the same phenotype. And indeed, you can see that if you look on RUNX knockout hips, you can see that there is a complete dislocation of the hip in this case, and a lot of malformation, as you can see here in the quantification of the different measurements. So just to summarize that part, I showed you that piezo-2 loss of function in the proprioceptive system result in skeleton abnormalities. I showed you that this finding can explain the human piezo mutant skeletal phenotype. I once again made the point that the proprioceptive system play a key role in spine alignment and joint development, and that this finding further support the role of proprioceptive system in skeleton biology, and basically support our greater hypothesis that the proprioception might play a role in many other syndromes in the musculoskeleton. And now we are finishing our analysis for identifying variety of genes that we think can serve as targets, and we're going to look now not only on the skeleton, but also on muscle and tendons as well, to see if there is other problems, and hopefully understanding better the mechanism underlying that. Finally, I want to thank the people who did the work, and my group, and also our funding, and that's it basically.
Thank you so